Welcome to Diagnosing Dx, the podcast giving diagnostics the platform it deserves. While therapeutics and devices often take centre stage, diagnostics is the engine driving precision medicine. I'm your host, David Philby, and each week we spotlight the innovators, breakthroughs, and real-world challenges shaping the future of DX. In this episode of Diagnosing DX, I'm joined by Josh Smith, founder of Bench in a Box. We dive into the breakthrough moment that inspired his platform, how it strips diagnostics down to their essentials for use anywhere, and the challenges of fundraising as an early stage founder. Josh also shares his perspective on what makes a scientific founder truly investable. This is a fascinating look at diagnostics innovation at the very beginning of its journey. Welcome to the show, Josh. How are you?
Good. You know, it's Monday, so looking forward to getting things done this week, but
Yeah. And
had
where
my
in the world
coffee.
are you?
The space in the thumb of Michigan. So
Nice
Bay
time of year
City.
to be in Michigan. Yeah, it's Michigan.
Yeah.
It's summer in Michigan. You can't beat it.
Well, you know, we just actually had our first night that dropped into the 50s, so I'm quite happy with that.
But then that's a slippery slope to winter. So
Yeah. Well, I come from Wisconsin. Michigan's got nothing on it so
yeah.
for winter.
Well, thank you for coming on the show. I've been excited to talk because actually, where you are as a company and as a product is quite interesting, quite unique in terms of we're really quite early. So actually, being able to talk to you as a founder at this stage, I think it's going to be super interesting to hear what you're having to go through and how you're doing that to really try and progress things. So before we get into what we have planned, it'd be great to sort of know a bit more about you and your background, what you've been doing in your career.
Yeah. So I am I have degrees in genetics and development and graduate degree clinical microbiology. I've been in labs. Oh God, I even started in my freshman year doing more than just washing dishes. I had a brand new professor that was trying to get stuff done, and I got lucky. So I've actually been doing actual bench research for 20 plus years, presenting and all that. My first job out of grad school, as pertains to the company, is I was working at the University of Wisconsin Biotech Center. And while I was there, my entire job was to set up basically a bespoke assay service. Real-time PCR was just coming out, and it was a gene chip lab. So people would go on the fishing expeditions with gene chips to see what's upregulated, what's downregulated, you know, that something's staying the same, something's not there. And they would come to me, and I would build, at the time, RT-PCR, so reverse transcription real-time PCR assays, on an old Roche 480. And they basically come to me, give me the gene they want to look at. I'd build the whole assay for them, run the experiments, give them the results, and they pay us for it. After that, I worked at a pharmaceutical R&D company, and between building some surveillance assays for the products we were going through, but we had one that made it through 1A clinical trials, and the other was some preclinicals, but it was basically using E. coli as a mechanism to fight infections rather than a normal, say, antibiotic. So we stripped out a lot of, they were pretty, well, if you looked at them, they died. So they were pretty non-pathogenic to begin with, but it used bacterial conjugation, which you can't defend against, and there's not really any mechanism for because it's a passive thing. Worked really well. We had a lot that we were working with the DoD for. And then after that, that was a startup too as well. But I started and I branched out. I started building in a cannabis testing facility. So it was the product compliance testing. So heavy metals, pesticides, microbial contamination in order to be released. and we grew into bought out a second location in Detroit, and I was doing that until 2023, 2024. Was consulting for a little bit throughout there, and in the meantime, sat on a couple of committees, ASTM, AOAC, a couple of scientific advisory boards, and lobbied for a little while. Well, I tried to do the all-inclusive for the CEO of a startup was, you know, if somebody's gonna stab you in the back from the legislature, you at least want to see it coming. So, didn't I was there enough to do the lobbying, but for most part, it was really me keep an eye on any competitors that were trying to do anything or any people that were trying to basically kill off stuff at the beginning. And then I was at a Department of Defense uh, contractors trade show, the JSTOR down in Florida, and I was walking around the posters and something, I don't even remember which one it was I was at, but something clicked for me about a product that I'd kind of put together years previously. And it just, that last little piece of how to turn it into a full diagnostic platform straight away kind of hit. And so that's where Bench in a Box comes from.
Okay, so we're getting into it. So, um, what was it? Was that your first? Let's talk about diagnostics
Mhm.
specifically. Throughout your career, where's the sort of major emphasis in diagnostics as an industry? Because obviously you've been involved in a few different things, right? So
Mhm.
has there been a sort of diagnostics thread throughout, and it's all sort of coming together now as a founder, or is it more recent, or is it the early part of your career
Oh,
and now later now as well?
it's a little bit between the whole time, and I mean, there's definitely a lot more in the first 10 years being in
Yeah,
pharma. But real-time PCR, so qPCR, that's kind
yeah.
of been my big where I spent most of my time um, and built through. So even uh, at my own previous startup lab, I was building different real-time PCR assays while
Mm,
there. Um, one for gender testing of different plant species or of different plants, some just minor bacterial or microbial contamination ones, trying to split between different aspergillus species that were coming up, that kind of thing. So
mm,
and I've also kind of helped consult with some people on the side too, specifically in the diagnostic side of building that usually real-time PCR assay or helping them figure out ways to take their base assay that they want and expand it to be used for more things than just what they're looking for. Because what most
yeah.
people don't realize is a lot of times yeah, you can plug the stuff into like IDT's calculator or wherever, and they'll give you a primer set. But depending on where you pick that primer set, you can get it to tell you a lot more stuff. For example, a friend of mine was using, she was working at the University of Wisconsin Hospital, and they were testing and surveying senior populations for Clostridium difficile. And the assay that they were using, one, the person trying to make it was taking forever. It was like 8 months in. But I, by just tweaking where you put that primer set, you could actually then differentiate between the two or three major types that they were looking for, not just saying, hey, C. diff is here. And it's
Mm.
things like that, which, you know, from being around at the beginning of that kind of boom in the industry, we looked at a lot more things, whereas now it's more just put it into the calculator or put it in the software and have it come up. And sometimes it is faster and easier. I'm not, you
Mm.
know, every once in a while you just want to get it done and get it rolling.
And you said you had an initial idea
Hmm.
Where did the initial idea come from? And I think you said that you sort of sat on it for a little while, didn't know how best to place it. But where did that initial idea come from, and how was that formed before you started the company?
To be honest, a big chunk of it came back in grad school. So we had been working on this molecule. It was HPMA. It was from Proteus mirabilis, and it was an interesting compound. It's a protein that's just weirdly shaped. It's just like a tube, it's a three-sided tube, and it's hemolytic. It's a hemolysis. But during grad school, we had found that, or I had found that if you, well, my professor did, sorry, let me back that up. My professor found that if you had truncated it and had it properly secreted and folded, if you had the full length, non properly secreted form, so non functional, non folded form, if you put them in together, it refolded. So we were doing, or not, we were looking for if different cysteines or sulfur compounds or different metals changed that binding affinity, and we were looking to see if there was an actual activity mechanism of that protein, and it wasn't. It just seemed to be very much structure based, kind of like prions. How like prions, if you put them in, they're actually improperly folded proteins, but they're much more stable than the ones that are found in your brain. So when you put them in contact with each other, the much more stable one almost forces the one that you normally have functioning in your body to the more stable, non-functional form, and then that's where that plaques and those holes start coming from. It's kind of terrifying that it's just as stable as they are. But so I built several kinds of assays looking for full-on detection of this and kind of kinetics and building it out. And it just kind of sat for a while. I always wanted to do more with it, and I couldn't think of ways to do, just that last little bit. And then, like I said, this past year, when I was walking around the show, I actually figured out how to attach targeting mechanisms to it. Because, and this assay runs in 5 minutes, roughly, when you put it in at room temperature, and it's exponential like qPCR, but there's no enzymes in it. It's just the proteins and your targeting molecules.
Hmm. And so you have this breakthrough moment walking around
Hmm.
a conference, but that's not the same as starting a company. So,
Correct.
who do I call you? Biab Tech? Do I call you Bench in a Box? Do I call you Bench in a Box? So you tell me, like, what, and tell me
Imagine
who you are. At Alevra Consulting, we focus on DX only, supporting early stage and growth companies achieve their milestones by solving their biggest talent challenges. Have you ever suffered from working with a talent partner who was either life sciences or med tech and dabbling in diagnostics? Or no more? Work with a diagnostics specialist talent partner.
bench in a box. If bench in a box technologies gets a little long, the reason why I even chose bench in a box was I during my education I did some rotations slash residencies at hospitals and hospital microbe labs, and you'd go to the urine bench, you'd go to the stool bench, you'd go to the wound bench, and that's just kind of where that
Yeah.
thinking came in is, oh well, this is, and then you just put that in a little box, and that's your testing thing, and marketing. So, but yeah, startup is kind of where I decided. I was like, well, now that I have that last piece, let's go for it. Let's pull the trigger
Yeah.
on it.
So
I've got the proof of concept already done and built, and it works astoundingly well. In some experiments, I've gotten it down to as small as 3 minutes for
wow.
results, and we're talking nanogram amounts or picogram amounts of template.
So talk me through what the product's actually going to do then, and the purpose and impact of the product. What's the problem you're trying to fix, and how's the product going to do that?
So anybody that has worked in molecular diagnostics or anybody that has done most kinds of diagnostics actually knows that you need a lab bench for it, you need space for it, you need to train somebody how to run it, and most of the time you need freezers, equipment. I mean, what's a I think thermocycler's list price for a 5 or 6 or somewhere like 40 grand at that point. If for you can get it cheaper, um, but even ELISAs, you still got to buy the antibodies and the stuff to go in it, and you've got to keep it cold for the most part, um, or at least the components of it. So I come from a pretty rural area of northwestern Pennsylvania, and throughout my years, I've been to places and seen places that are very, or even more remote and rural, and have a lot harder time with medical and clinics and being anywhere near people. I also, my family, if I like to say, if my father didn't have fever, I would have been the seventh generation of the same dairy farm. So I've grown up around agriculture for a while too. And what Bench in a Box does is it strips out all the non-essential things. It basically strips your PCR down to a bare bones thing. Just protein, your target, and like a binding molecule, and then a dye reporter. So you can take it to places like those rural clinics. You can take it to disaster relief areas. You can take it to, fields for veterinary, and you can get that same kind of advanced diagnostic capabilities without any training and where at its points that it's needed rather than having to collect the sample, ship it off to the lab or ship it off to the clinic. I like to say, imagine getting when you walk into the hospital or your clinic and you get a test for RSV, influenza A, B, strep, staph, COVID, and a whooping cough and any other things in the same amount of time as a rapid strip, all in on one test.
Wow. And so you mentioned earlier in terms of having a prototype, or it's where's the product up to, and where are you as a company now then?
So proof of concept's made. We're looking for our MVP. That's what our current fundraise is for, is to actually go through and build, because the vectors and things that it's built on aren't necessarily made to be high throughput or like for mass scalable manufacturing. Um, and then we're also, I've been in talks with a company to look in, and they're interested in expanding some products that they're offering. But I've been looking into partnering with them to build microfluidic chips, bespoke microfluidic chips, made out of like quartz, so that they're a little more rugged. Um, and that's the actual long-term goal, is to get it into a microfluidic MVP product that we can ship. Originally, we're going to be looking at the veterinary world just because of regulatory pathway and ease to market.
Yeah.
Specifically, I'm looking at like bovine TB um, and canine heartworm testing. Because the nice thing about our platform is it doesn't just rely on nucleic acid nucleic acid oligonucleotide probe. You can use any kind of thing intact to our protein reporter, that anything it targets, anything that binds. So antibodies are also included in that. So you can widely expand any of your targets and not just require it to be nucleic acids.
Mm. So you target the uh veterinary world, because of the less regulation, it's easier to get to market. If you get to market, how quickly do you then look at pivoting back into sort of healthcare, human healthcare?
Um, the goal is to, at that point, start setting up divisions and then going all in on both veterinary and clinical.
Yeah.
And also, there's environmental screening, so like water screening for E. coli, salmonella, that kind of thing too, is another one we'd like to drop in there too.
So you mentioned your fundraising. This fundraise is ultimately to get to MVP.
Hmm.
What's your perspective on fund? Because I'll tell you why I asked this question for context. Often we talk to, especially on the show, I talk to a lot of founders in terms of they're in the same boat as you, but on the show we would tend to have somebody who's come on who's probably just hit a milestone and wants
Hmm.
to sort of share that, and that's very absolutely fair. You're knee deep in it. How do you approach that, and how do you how is how is that at the moment? What's the feeling you're getting from the conversations you're having for raising funds at the moment?
So the last fundraise I was involved in happened well, we opened the fundraising round in January of 2020.
Okay.
So, so everybody,
Yep.
for everybody that didn't put that connection together, COVID hit and all the lockdowns began. So, um, that was much more, that was very difficult. So
Oh,
that's
so
my,
you've got some good experience, and this
yeah.
is
So I've got some great experience at, well, damn near impossibility at that point.
yeah,
So, but it is tough. I mean, especially in the US right now with the way the economy is sitting and the way interest rates are sitting at, it's a little bit of a harder sell. You've got to prove to people that you actually, the team's there, that you have something. It's not just that pipe dream kind of thing. Um, from what I've been hearing, experience of your team or your founders is kind of a big deal. Not just being, the brand new guys fresh out of college are going to be a little harder to get on than somebody who's been through it a few more times.
sure.
Um, but I think the amount that people are willing to part with in the early stages is a little lower. So chances are people like me are going to have to talk to a few more investors and get a few more to sign on first. But I think it's it seems like it's still there and it's still doable and there's people still looking for returns. I think it really just depends on who you're talking to. If you have somebody are you just looking for the general angel, or are you looking for somebody that specifically deals in diagnostics or pharmaceuticals or whatever it is that you're trying to do? Because they're going to understand at least where you're coming from more than just those general guys. Um,
Mm,
and yeah, it's tough, but it's still not impossible. It's not impossible. It's just you've got to be a little tighter on your message. You've got to be a little tighter on who you're talking to, and you really need to make sure that you can justify what you're asking.
yeah. Have have you put together a founding team or advisory team? Because you mentioned that's one of the ways you're trying to stand out, and obviously your own experience versus someone who's
Hmm.
coming straight out of academics. That as well. But what else are you doing to sort of try and stand out as a company in the eyes of investors?
So one of the things I found in the last startup, the lab, that made it kind of difficult was I didn't really. It was mostly just me when I was doing back then. I luckily had a couple of guys that were willing to put money on what I was selling,
Mm,
but But this time I've really spent some time kind of shoring up where those what I saw as deficiencies or pain points that seem to be coming up. So I put together uh, a full advisory board that most likely will become more than just an advisory board uh, pretty soon. Um, but I've got a guy that has been running pharmaceutical startups for quite a while. Dr. Steve Watt. He actually was my former boss at the pharmaceutical company. He's
Mm.
still there. Um, but he's got dealing with governments, government contracting, the FDA. He kind of covers that side of things. Um, then I've got Evan Friedman. He's the vice president of Helma USA, which is a huge scientific supply company. Specifically, they look at the glass cuvettes and quartz cuvettes for spectrophotometry. But he does logistics and setting up like a actual supply company. And then Nathan Johnson, he's at Vern Bio. He's multiple companies at this point, but he's molecular diagnostics from the. So he's done that before. So it was
Mm.
kind of trying to get all of those things that I've heard or over the years people have mentioned, well, do you have anybody, on staff that's for this or for this or for this? So that's kind of where I came about looking to do,
Mm.
or at least, you know, step up and address those before they even became questions.
Yeah, makes sense. Let me ask you this as a founder with a rich scientific background, and I know you've got a good sense of humor. So, how do you, as the face of Bench in a Box, ensure that you're not seen as the crazy scientist who's in love with their product and actually going to be capable of building a business, which is what investors want to see? And you know this, we see this so often in the industry of, yeah, but it's the business bit that we missed, and so how do you
Hmm.
prepare for that, and how do you sort of get yourself, when you're going into those conversations and processes, what do you have to do to be able to see as an investable founder?
Um, it's confidence, right? They've got to be confident in you that you can actually pull this off one way or another. I mean, unfortunately, anybody who's in biotech, whether it's diagnostics, pharmaceuticals, or any other kind of biotech, uh, we have the unfortunate problem of we need money up front. We need it, like, we can't do much without equipment or
Yeah.
without stuff. So we're a higher ask right up front than people are used to, say, from the tech industry and all that. So, like I said, you you have to instill that confidence in them. One, that you can do it, but I also think too that you're willing and able to accept feedback, because from my own experience talking to founders and from things I've read up quite a bit and tried, or at least tried to keep abreast and try to sneak into places where I get comments from people. A
Mm.
lot of the times founders have been drinking their own Kool-Aid so much that they can't accept that there are different ways to do things, or that there are holes in what they're
Mm.
bring to the table. So, like, for example, um, I not a marketer. I can do the CEO thing where I can go to a place and I can do the high level kind of, hey, we could do this for you, and I can get in the
Mhm.
lead. But the actual salesperson closing, having that pulse or the feel of the pulse of what marketing is going to work, that's just not me. I'm a lab guy. I, so I've been currently my list of tasks has been trying to find someone to go add to my advisory board that's a marketer. I had one didn't that I liked. We talked conflict of interest. It didn't pan out, so I'm still currently digging around for it. But like, also scientific supply company logistics. That's part of the reason why I asked Evan to join, was because once you get past that, I got to find a way to actually ship, whether it's national, international, or whatever. You're going to have to kind of have that in place, and somebody that's already been doing that for a while is great. Any other little products in hand. So it's really, I think you need to be brutally honest with yourself as a person and
Mm,
what your strengths are, and having the ability to just to address them honestly. Because if from or every investor that I've ever spoken to, that when I ask the or when it comes up, it's like they know there's going to be holes in something. Like
yeah,
no plan is perfect.
yeah,
Just even like competitors, like that's a big one that I've always seen is no, you have competitors. Whether or not it's the same exact thing as you or not, you always have competitors. And if you don't, that's actually a bigger problem. But you being able to honestly address them and say, this is why we beat them. They're going to be, most of them are going to beat you in scale. Like they can whip something out with development or with just sheer monetary costs. They're going to beat you. So how do you address that? How, or if they have 50 patent attorneys on file, kind of a thing. They get, somebody's going to get around. And one comment that I always heard was, whether it's a patent or whether it's an NDA, it's only as good as the money you have to enforce it.
I love that. Yeah,
But I really, that the whole thing that's the big one for me, from what I've seen. And you can get into strategies and have an actual plan, and you having a budget that you actually makes sense. Like you don't have to go into the full nitty gritty details. Some of them want it, but for the most part, they just want to make sure that you're actually doing it. But you can take feedback and you can take advice, because most VCs, let's face it, they're going to want to see it on your board, and they're going to be able to help steer the direction of things when they give you
yeah,
your money.
yeah.
So you got to be able to. I used to coach ice hockey, and one of the biggest things I always got asked was, "Are they coachable?"
Mm.
Kind of same thing. Uh, Resonates, I guess. It's not exactly the same, but it's close enough. Can you
I
take
think I
that
think it's
thing?
yeah,
Yeah.
yeah. I think you're right. I love the at the start of your breakdown of that was really humility,
Hmm.
humility, and sort of knowing what your strong points are, accepting. And actually, I love the point that you make about investors will know there's holes. Don't try and preach perfection. That's worse.
Hmm.
Instead, embrace the gaps and how you're going to address them. Because we get this, or where it goes wrong is you either have, if it's a scientific founder, you have the complete lack of awareness commercially, and therefore just in love with the science, right? And
Hmm.
this is going to change the world, but actually what they think will change the world doesn't mean it'll be successful as a business.
Correct.
Or you have the opposite, in that you have a scientific founder who thinks they know better than the commercial people they get around them, and it's like, yeah, give them the space and freedom, and like you say, take that feedback on. So I loved how you said that. You also
One of
you meant, go on, Josh.
Oh, I was just going to say, one of the so in grad school, you you take your oral exams, right? And at least where I was, they one of the biggest lessons, not just that you learned the stuff that they're asking you, was that they went until you literally blanked, like your brain blanked. And the whole point, or the secondary point of this whole process, at least for my advisor, was to get me to say, I don't know. Like, out of everything. Because if I tried to start making stuff up on the fly when I got to that point, they would just brutally, they would follow it out until you base the whole point. was to admit that you don't know it. You don't know any,
Mm,
like it's okay to admit that, but I think it's something that's kind of lost in some of the training anymore because, and maybe it's just the people that, always get on TV or whatever, or the
yeah,
interviews, but
yeah,
I think that as a whole is a great foundation for that kind of thing because we don't get the business, teach classes to learn how to do that. I mean,
yeah,
it's not hard, but um, so yeah, just to kind of bring that around. Sorry.
yeah. No, 100%. It makes sense. You mentioned competition. What does the um what does the landscape look like in terms of other platforms and products that are trying to disrupt the need for traditional PCR at the moment? And how does bench in a box fit into what's out there?
So the thing with especially like the PCR market is people are just building on that same structure, whether it's next gen or whether it's qPCR or isothermal PCR even. Um, it's still using that same basis of amplifying your DNA or making a lot of copies of that DNA, attacking your probe on it, and using that as a way to amplify your signal. Because at its base, PCR is just a DNA photocopier. So
Mm,
and then you're lighting it up. So what Bench in a Box is doing that's different is I'm not making a lot of copies of my template. I'm amplifying the signal on the back
mm,
half. So instead of so basically PCR does it up front, I do it on the second half. But that allows me to eliminate the need for enzymes. I don't need a thermocycler. I don't need a freezer. I can, it's strictly, at least right now it's colorimetric. So you can just, heck, you could probably even set up a phone app for it. I really don't like that. It always felt gimmicky to me, but you know,
mm,
if it works, it works. And then there's other things down the line that I'm interested in attempting, but at least for right now, just the main difference is simplicity. And I tried to boil it down to it's the signal we want. Why do we need everything else?
yeah. And is there other products like this in development at the moment that you know of, or even in commercialized?
So there's a few that I've had pop up. Um, and there's one out of, I believe it was MIT. Uh, they're using electrical signals on glass chips or on glass slides and some weird chemistry that to be honest, is way out of my chemistry depth. Um, they're looking for a different kind of targeting system, and it's more of an endpoint and just more of a general, like, oh, there's stuff there.
Mm,
But they're working at using that voltage difference to kind of show different contamination or different organisms or things. It was pretty interesting. I'll send you the link to it if I can dig it out if you're interested.
yeah,
So, but that's kind of they're all more in the middle phases. The only thing I would I personally think is close is isothermal, and that's using the primers themselves to kind of replicate themselves, but they still need that difference in temperature. I mean, you've got a anneal and uh, they separate the DNA strands from an actual just and everything. And the other stuff is mostly equipment based. So your Molotov, your uh, that's always the one, the Q-tops, the
mm,
Vitek systems, those kind of things.
yeah, makes
Showed my
sense.
age with saying Vitek.
And what does the future look like from here then in terms of upcoming milestones? And I think you mentioned you've had some good news recently as well. So what sort of
Yeah.
milestones should we look out for from Bench in a Box?
So we now, as of last week, can officially say patent pending. Finally got that locked down. Um,
Awesome.
and now we're really time to start hitting the fundraising trail hard. That was kind of the last piece that was I've been reaching out, but I really we all know that's a holdup. I mean,
Right.
you got to have it locked down. So
Right.
now that that's hit, it's really fundraising in earnest.
Congrats on the patent pending. That's awesome.
Thank you.
That must be good for you, a good feeling
Oh,
for you, right?
it did. It did. You know, there's always the okay, which claim, what are they going to like, what are they not, you know, and the whole back end. But it's definitely a weight off the shoulders.
How long did that process take?
So the initial part doesn't take a lot. Um, really, the first bit with like provisionals, you can get pretty quick. You want to make sure from other patents that I've had, um, they usually, yes, you can go by as quick as just slapping down a paragraph, put a drawing in, and then make your one claim,
Mm.
because nobody looks at it. But for the full size, they actually, if you're going to go that route, the patent attorneys and agents, they really do like you to actually have a full, or at least close to build out. So it takes a while. I would say, oof, probably good 2 months, 3 months,
Mm.
at least for to get through and get everything written. Because the hard part is going through all the prior art and then
Right.
flagging it and making sure that you've got a rebuttal as to why it's different. Um, and
Mm.
then making sure you've got sufficient background and finding ways to describe, and this is where the money's worth it for the patent to actually hire somebody that's been doing this, is you want to build your strategy of how you want to go about your IP wall. Because you don't want just one big patent that has everything in it,
Yeah.
because it's only good for, I think it's 11 years now.
Mm.
It's only good for that. But if you have everything in there, you can't tack on new ones and you can't extend your your security. So you've got to figure out, okay, what's more important for this one, what's not important, what's something that we don't have a lot of data on right now that but we can, so that we can build onto that for later time. So like
Right.
different detection methods based on your stuff, or
Right.
different platforms or things like that. So it's there's a lot of ways to do it. You just need to be conscious and of thinking through it and how you might want to play the long game.
Yeah. Makes sense. That's interesting. Obviously, I've never done it myself, so I'm actually genuinely interested in that and how that goes. Um, so you're now knee deep on the fundraising trail. What's your sort of expectation in terms of timelines that you're hoping to get this next fundraise closed?
Well, August is holiday time for everybody, so pretty much, don't expect a lot of
So you're not
traction in
doing
August.
anything right now.
Yeah,
just
but
just
I'm
take.
reaching
It's a good
out.
start.
Yeah. Yeah, I'm reaching out. I'm just not expecting to hear back from a lot of people since, if you
Yeah.
have most people that have money take a holiday in August. The same thing
Yeah.
like working with European countries and or anybody that's in a European country in December. I mean, don't expect much to get done in December. So it
100%.
it actually all of this kind of works out pretty well with getting. So while I was doing the reach outs, I wasn't too concerned about it. I was using this time to kind of get caught up on all my other stuff. We just got a redone website, an updated one from that single one page placeholder that I had previously. I've got the patent pending now, and just kind of getting everything ready to go so that you can hit the ground running. All the HR forms and things, getting all of your everything tight. Your operating agreements, making sure those are airtight, that kind of thing. Um,
Yeah.
so I'm looking to really kind of, God, I'd love to have this done by first of the year, but
Mm.
depending, it always depends on who you talk to and how the economy is
Yeah.
going. Personally, I'm going with it's this is the safer bet than some of the sectors of the U.S. economy. So
100%. Well, hopefully the landscape is a bit forgiving. We want more funding into early stage novel technologies, but unfortunately, investors want more proof of
Mhm.
concept and less risk, and that's just the oxymoron of the industry at the moment. But
it's right up there with getting that first job out of college. Everybody wants
yes,
experience, but nobody wants to give you
yeah,
experience.
it's
Banks
exactly that. That's
banks will give you money, but
fantastic.
only if you don't need it.
Right. So there we go. Well, look, Josh, this was fascinating, to be honest. I love being able to talk to you at this stage, and I'm excited. I'm rooting
Mhm.
for you. I'm super excited on the product, especially now that you have patent pending, and to see how this journey unravels over the next 6 months, let's say. Um, Anything else you wanted to add on bench in a box before we wrap this up?
Uh, no. The only thing I would just say to anybody that's listening, if you're, somebody that's stuck in my position of this, I would just say it's if you've never done it before, just know it's tough. It's a very difficult thing. You're trying to take something that you've got, maybe it's a one-off, maybe it's you've kind of putzed around with it in your lab, but it's worth it in the end if you can do it.
Yeah.
But just know that, the reason why everyone doesn't is because it is extremely difficult. So stick with it.
Yeah. Thank you for that, Josh. Josh, this is honestly awesome. I'm
Yeah.
I'm really rooting for you and bench in a box, and I'm excited to see what happens next. Thank you so much for coming on
Yeah.
the show.
Thanks for having me. It's been fun.